Hello students, I am Dr. Prerna Bhasin, consultant anthropologist and director in Spire Analytics. We are here to discuss a module on color blindness PTC tasting, which is a part of a bigger paper on research methods and fieldwork. We are going to understand the learning outcomes for today's module. We are going to appreciate that why anthropologists study human variation. We'll familiarize ourselves with the definition of color blindness, its type, the causes, the incidence, importance of color blindness screening, tests used for screening and calculation of the allele frequency. We'll then move on to discover the idea of PTC, the differential tasting, the taste sensitivity, genetics of PTC, association with other diseases, tests used to screen for PTC taste sensitivity, and procedure used to calculate the allele frequency of the following. As physical anthropologists, we are interested in studying human population. In this process, we try and reconstruct the history using material evidence and biomolecules recovered from the fossils. Another approach in understanding human evolution is investigating diversity in human populations. Earlier, anthropologists studied human diversity or variation using visible physical traits like skin, hair, eye color, hair form, shape of face, and nose. Advances in serology and molecular biology have made it possible to study blood groups and of late biomolecules like DNA, RNA, and proteins. Human variation arises due to operations of evolutionary forces like mutation and migration, also known as the gene flow, and besides that with genetic drift and natural selection. It was observed that the percentage of variation within and between population is 85% and 15% respectively. Now, study of human variation is not only important from the anthropological point of view, but is also helpful in understanding or giving us a perspective to understand the contribution of genetics to the human disease and long-term survival of human species. Human population genetics is a subfield of physical anthropology concerned with study of the allele frequencies of various genetic markers like blood group and also factors responsible for allele frequencies over a period of time. Now, color blindness and PTC are the best examples of population genetics. Color blindness, as you know it, is the fact that how light rays are reflected from the object that pass through the cornea and then pupil and lens and produce an upside down image of the retina. Now, figure 1 here will depict the structure of the eye, how retina converts light into visual signals and then transmits it to the brain with the help of two photoreceptors, namely rods and cones, where the image that is interpreted as upright. So, retina has 100 million rod photoreceptors and helps in the night vision, and it has about 6 million cone photoreceptors which are responsible for the daylight, color, and spatial vision. Normal vision is called trichromatic due to the presence of pigments of 3 types of cone receptors that are sensitive to the blue, the green, and the red wavelength, and they're all of different wavelengths. So this first is 420, second is 530, this last one is 560 nanometers of visible light. Among the 3 cone types, L and M cones are rich in fovea, that is, they are concentrated in the central part of the retina, while S cone that constitutes about 2 to 7% of the total cone population. Cone pigments are called opsins. They are composed of 340 to 370 amino acid residues. They folded into 7 membrane structures and then embedded in the outer segment of the membrane. But the focus here is on the color blindness. How and why does it occur? Color blindness is coded by a gene, and the gene for S cone opsin is located on chromosome 7, Q 31st region point 3 to Q32, whereas gene for L opsin and M opsin are located on X chromosome, Q region 28. in a head to tail tandem array. The ability to appreciate color distinguishes humans from non-primates. Color blindness or color vision deficiency is defined as the inability or decreased ability to perceive color under normal lighting conditions. What are the types of color blindness that we need to know about? The first type of color blindness that you need to know is achromatopsia. Persons with achromatopsia are characterized by reduced visual acuity, pendular nystagmus, photophobia, small central scotoma, and eccentric fixation, reduced or complete loss of color discrimination. Incomplete achromatopsia, impaired color discrimination, and lack of function in all three cone types is observed. In the incomplete form, afflicted person may have partial functioning of one or more cone types and may have less severe symptoms than individuals with its complete form. Monochromacy. Typical monochromacy, it is caused by mutations in the gene encoding the cone-specific alpha and beta subunits of the cation channel. Typical characteristic features of persons with this type of color blindness are nystagmus, that is involuntary shaking of the eye, differentiation of color by brightness, photophobia, insensitivity to red light, and low visual acuity. What is blue cone monochromacy? This is the second type, and this type of color blindness is characterized by presence of S, that's the blue cone, but absence of L or M, that are red and green cones, differentiation of color by brightness, rudimentary color vision in mesoscopic vision from rod, and cone activation. There is also photophobia, nystagmus, insensitivity to red light, and low visual acuity in the affected people. Let's understand what dichromacy is. The first type is protanopia, that's red in color. In the affected people with this type of color blindness, they have no L cone pigment, a confused red, yellow and green, white and green, and blue and purple, and reduced sensitivity to red light. The second type is deuteranopia, that is green in color. This type of color blindness is characterized by the absence of M, that's the green cone pigment, confused red, yellow and green, and white and green. The last one is trinatopia, that's for the blue one. In persons with trinatopia, S cone pigment is absent. So they confuse blue with blue-green and white with yellows. The next figure highlights the visual perception of a normal and a colorblind individual. Let's try and understand what anomalous trichomacy is. The first type is protanomaly. Characteristic feature of this anomaly is shifting of L cone pigment absorption spectrum to a shorter wavelength. So that's like going from red to blue cone pigment. Afflicted person confuse red, yellow and green. There is reduced sensitivity to red light, abnormal color matching, and then they add excess red in the color match, that is red plus green, that will appear yellow to them. There is deuteranomaly. In this type of anomaly, shifting of M cone pigment absorption spectrum to a longer wavelength, so going from green to maybe red is observed. Furthermore, they confuse white with the greens and reds with yellows and greens. They match abnormal colors together. So adding excess red in the color match, like red plus green, will appear yellow to them. Trinatomaly. In this anomaly, partial loss of S cones is observed and also failure in discriminating blues, blue, greens and greens. The next figure indicates difference in color perception in different colorblind conditions. While we are studying color blindness, it is important to understand what causes color blindness. What's the etiology of this? The etiology of color blindness is either genetics or it is acquired. The acquired, that is disturbance occurring on visual pathway from photoreceptors to the cortex causes of color blindness may include damage to the eye, the nerves, the brain, or it may be due to diseases like diabetes, glaucoma, hypertension, macular degeneration, It can be optic atrophy or optic neuritis, and even sickle cell anemia is known to cause this. Exposure to certain drugs like anti-tuberculosis drugs or barbiturates is also known to cause some of these color blindness types. Epidemiologically speaking, we also need to understand the incidence of color blindness. Among all color blindness types, frequency of red-green color blindness is known to be highest. It was observed that in higher proportions in males than in females. Highest frequency of this color defect was reported from Northern Europe, that is 1 in 12 males and 1 in 200 females. Blue cone monochromacy frequency is also found to be higher in males than in females. The incidence of blue cone monochromacy was reported as 1 in 100,000 people in the world. Blue-yellow color blindness was observed equally in males and females. The observed incidence of blue-yellow was 1 in 10,000 populations. The next figure indicates the inheritance pattern of the color blindness. The importance of color blindness screening comes from the fact that apart from using this as a tool to study population variation, color blindness is of socioeconomic importance as well. While driving vehicles, recognition of color of traffic lights and color of articles of daily use in shopping is essential. Various occupations demand color recognition, and these occupations, those without color vision defects, can be productive and successful. For jobs such as deck and navigational watch keeping in maritime industries, pilots in aviation industries, train drivers and workers in railways, drivers in road transport, aircraft pilots in Air Force, officers and deck crew in Navy, traffic police, firefighters in fire and rescue services, technicians and linemen in telecommunication, and electrician individuals with color vision defects are not preferred for these jobs. In some jobs such as technicians in diagnostics, pathologists, dentists, optometrists, horticulturists, fruitiers, architect, interior designers or decorators, graphic artists and fashion designers can be at disadvantage because of the color vision defect. How do you screen for color blindness? Color blindness is not curable. Color vision deficiency can be managed to some extent by using adaptive strategies. Informed choice can be made in careers, and disappointments can be avoided later. This is only possible if screening is done at an early stage of life. Color vision defect can be diagnosed by using 18 different types of pseudo-isochromatic plate test, 8 sorting arrangement test, 5 anomaloscopes and 5 lantern tests. No single test provides all the information on the color vision defect. The ideal test should be easily available. It's a low cost, administered quickly, interpreted very easily, and a reliable form of it. Now 4 tests such as Ishihara test, Richmond, the HRR test and Medmont C100 test along with the Farnsworth T15 are used for detection of color vision defects. Among all these tests that I've just said, Ishihara test is widely used. It requires little guidance for administration and interpretation. It was extensively used by anthropologists for detection of color blindness in various populations. Therefore, it's time to understand the Ishihara test. Ishihara test was designed by Shinobu Ishihara, who served as a surgeon in Japanese Army and was later appointed as the professor of ophthalmology in the University of Tokyo. At the International Congress of Ophthalmology held in Holland in the year 1929, it was recommended that the army would be tested for color blindness. The test is available in 38 plate edition, abridged 24 plate edition and a 14 plate edition as well. Each plate has a figure printed in color dots of varying sizes against a background of dots printed in different colors. Colors are chosen to ensure that colorblind people either do not read or misread this. Now this test detects proton and deuteron with high sensitivity and specificity. Disadvantages of this test are that it cannot differentiate proton and deuteron in 30 to 40% of cases. They fail to report the severity of the color vision defect and are unable to diagnose a tightened color vision defect. The next figure exhibits Ishihara color vision test plates used for screening color blindness and then depicts Ishihara test for color blindness. You can also see the number and the one that is meant for color blindness detection among literates, and then there are winding lines to be used for color blindness detection of the non-literates. What is the procedure that we follow? So when your subjects are lettered, the Ishihara plates are held at a 75 cm distance from the subject. The plates are lit by daylight fluorescent lamps. So the color temperature would be around 6500 and the color rendering index is usually kept at 90 or higher. Subjects is requested to read the numbers in plates from 1 to 25 within the first 3 seconds. The numerals reported by the subject is compared to the standard table of Ishihara for detecting color vision defect. If the subject is able to read the numerals in 17 or more plates, he or she is considered as having a normal color vision. If subject is able to read numerals in equivalent or less than 13 plates, he or she is considered to have a color vision defect. There is description of Ishihara plates in the given table. So how do we help the non-literate people? The unlettered people or the person is requested to trace the winding line between two X's on the plate with soft brush within 10 seconds. Detection of normal and color vision defect can be done on the basis of the tracing of the wind line by following a criteria described by Bhasin and Chehel in their work in 1996. The next table indicates the difference in the color vision of a normal and a colorblind person by tracing winding lines in Ishihara color plates. You are now going to understand how to calculate allele frequency for color blindness. Let's look at this question. Shah et al. In their study in 2013 found that prevalence of 48 colorblind people in a total of 534 males and 12 colorblind females in a total of 590 women. There are 12 colorblind women in total of 590 women. That the allele frequency can be calculated in the following manner. So if colorblindness person designated is small c is 48 and normal vision person designated are capital C for 486, then the allele frequency for colorblindness should be 48 on 534, that's like 0.089 for men. And then the allele frequency for normal vision would be just subtract this number from 1 and then that will be 0.911. Similarly, if we do this for the women, we will find that the allele frequency for color blindness in women is, as calculated by multiple researchers, was found to be 0.121 as in the given formula. Combining these allele frequencies for normal vision, we found the number to be 0.86. Now that we have finished understanding color blindness, let's try and understand what PTC stands for. PTC is phenyl thiocarbamide. After the blood group, the widely studied genetic marker in human population is the sensitivity to the PTC. We exhibit here the chemical structure of phenyl thiocarbamide or PTC. In the pre-dawn age of molecular biology, PTC was used for settling paternity disputes and also considered as an honorary blood group. PTC was synthesized by Arthur Fox initially for making a dye in 1931. Dimorphism in the taste sensitivity to PTC in humans was discovered accidentally. Fox was pouring the chemical in the bottle to store it. Accidentally, a co-worker came in to exposure to this particular chemical, who complained of the bitter taste of the chemical, while Fox found the chemical absolutely tasteless. The observation triggered many studies on the trait of taste sensitivity to PTC in different human populations. We tend to depict this in the next figure, where the individual differences in taste buds density is shown. The next depiction shows the PTC taste test strips available in the market to test PTC taste sensitivity. Now, the human taste is of 5 types: the bitter, the sweet, the sour, salt, and umami. Taste sensitivity to PTC is a phenotype which has genetic, epidemiological, evolutionary interest and is also correlated with the food preference. So, tasting naturally occurs, something can taste naturally bitter and then you can avoid it. There are nutritional, survival and susceptibility to disease effects to the phenomena as well. Now, this whole idea of PTC comes from the L5-vinyl-2-thiooxazoliotone, a natural analog to PTC, was discovered in the cabbage and rapeseed and later identified as a causative agent for even goiter. Vegetables like cabbage, cauliflower belonging to the Brassicaceae family contains high level of glucosinolates which can break and give rise to isothiocyanates. These isothiocyanates were found to interfere with the uptake of iodine by the thyroid gland. Tastes avoid vegetables because a bitter taste would deprive them of a trace element. It was observed that vegetables like broccoli, Brussels sprouts and cauliflower taste bitter to tasters, and because of this reason they avoid consuming it, which also deprives them of isothiocyanates which are abundant in these vegetables, and these were proven to have anti-inflammatory and anti-cancer properties. Now, because of bitter taste of the medicine, they are avoided by children. That may land them into health troubles as well. Selective disadvantage among homozygous tasters in third and fourth decade of life due to hypothyroidism was proposed. Up till the puberty, no difference in the taste sensitivity was found in both sexes. After puberty though, comparatively more males were found to be non-tasters, whereas more females were tasters. Low threshold sensitivity to PTC was observed among women than in men. In one study, it was observed that 15% of the subjects who were non-tasters to PTC at sea level tasted the bitterness of PTC at a high altitude. It was hypothesized that high altitude hypoxia changes the hormonal profile and alters the sensitivity to the taste of PTC, resulting in some of the individuals shifting to lower PTC sensitivity. Identical twins were found to be similar in the perception of bitter compounds. Sensitivity to PTC decreases with age. Smoking before testing, mental status, ingestion of chemicals, radiation, metabolic disturbances, menstrual cycle, ear infection and head injury are reported to cause variability in the sensitivity to PTC. Another chemical used to test the taste sensitivity was 6N-propylthiouracil. Due to the lack of sulfur odor of PTC and its usage as a medication of Graves' disease. The bitter sensitivity of PTC and 6N-propylthiouracil was found to be due to the presence of a thiocyanate moiety, that's like with nitrogen, carbon and sulfur. PTC was found to bind more strongly with reported lower threshold sensitivity than 6N-propylthiouracil. With respect to tasting PTC, humans are classified as just tasters and non-tasters. Generally, individuals with 2 recessive alleles are termed as non-tasters, and people with 1 dominant allele or 2 dominant alleles are considered to be tasters for PTC. Incomplete dominance, 2 locus and polygenic models are reported in regard to the inheritance pattern of tasting sensitivity to PTC. Inability to taste PTC was suggested due to the failure of active G protein. Smokers, coffee or tea drinkers tend to be non-tasters due to the presence of bitter compounds which compete for the PTC. R.A. Fisher proposed the hypothesis that balanced natural selection maintains the taster and non-taster alleles in human populations. And further, evidence of biomodal distribution of tasters and non-taster alleles was found in the chimpanzee and orangutan. Wooding et al. Had observed that PTC alleles conferring non-taster alleles evolved separately in humans and apes. In Brazilian Indians, no non-tasters were observed. Frequency of non-tasters were found to be 25 to 57% in Caucasians and from 24.5 to 71.5% in Indian populations. Whereas the frequency of tasters range from 10 to 98% in the world population. In India, high frequency of tasters were observed in island populations, North and South India, whereas low frequencies were found in Central India and tribal population. In terms of proportions, the frequency of taster population in India was reported to be 42% to 66%. The genetics of phenylthiocarbamide is pretty simple. The bitter sense is experienced due to the presence of bitter receptors located on the surface of the tasting cells of tongue. Binding of bitter substances to these receptors initiates neural signaling while activation of G proteins. Receptor for bitter taste response is encoded by gene TSA2R38 belonging to the family of bitter taste receptor genes consisting of 25 members. These members have 25 to 89% of identical amino acid sequence. The major locus of TSA2R38 gene is on the chromosome 7, arm Q, region 35 to Q36, and secondary locus is identified on chromosome 16p. It has a single coding exon at 1002 base pair length and encodes 7 transmembrane domain G protein coupled receptor with 333 amino acid long. TAS2R38 receptor do not recognize compounds that lack thiourea moiety. We exhibit the main gene controlling PTC taste sensitivity through, that is the TAS2R38 gene. What is this association of PTC taste sensitivity with other diseases? Higher proportion of adenomatous goiter, nodular goiter, congenital atheriotic cretinism, dental caries, cystic fibrosis, schizophrenia, idiopathic and symptomatic epilepsy was observed in non-tasters as to PTC. Goiter, diabetes, mucoviscoidosis, duodenal and gastric ulcers were shown to be associated with the ability to taste PTC. How do we screen for a PTC taste sensitivity? Subjects are screened for PTC taste sensitivity using threshold and super threshold methods. In the former method, the lowest concentration at which the subject is able to taste PTC is detected, whereas in the later methods employ rating scales to assess the taste intensity at higher concentrations. The limitations of threshold methods are that they fail to detect taste intensity for higher concentrations of PTC. They cannot distinguish between medium tasters and super tasters, and non-tasters perceive higher concentration of PTC as intense as perceived by the taster itself. In super threshold method, multiple samples were used to detect taste intensity over concentrations or single samples for screening as point estimates of taste intensity or ratings of PTC, or the 6 and propylthiouracil were compared to other stimulants like sodium chloride or audible tones. Disadvantages of super threshold methods were found to be varying in sensitivity in detecting the taste intensity. The most widely used method for screening PTC taste sensitivity is threshold method as reported by Harris and Calamus. So how do you taste PTC and how do you test for PTC taste sensitivity? The principle is very simple. Depending on the genetic makeup of the subject, they would taste PTC either bitter or as tasteless. A PTC solution of 0.13% volume, weight by volume in double distilled water is prepared. 1.3 gram of PTC is dissolved in 1 liter of double distilled water. 14 dilutions are prepared by diluting the stock solution as shown in the given table. The stock solution can be prepared for 100 milliliters also depending on the requirement and number of subjects that are investigated. The subjects who are participating in the taste sensitivity testing are asked to rinse the mouth with double distilled water. The last solution is designated as solution 1 and the strong solution, that is the stock solution, is labeled as solution 14. Each subject is instructed to consume 4 ml of each solution. After each solution testing, subject may be instructed to rinse their mouth with double distilled water. After testing 14 solutions for taste sensitivity, if subject is unable to report no taste, he or she is labeled as a non-taster. In case of tasters, the solution at which he or she reports the bitter taste is recorded for determination of taste threshold. The allele frequency, that is taster and of non-taster, and the heterozygosity is calculated using the Hardy-Weinberg method. Now, if you see along, you will see the calculation for allele frequency or PTC taste sensitivity. In order to explain the calculation of allele frequencies of tasters and non-tasters, a study on the taste sensitivity of PTC is taken as an example. To explain the calculation of allele frequencies of tasters and non-tasters, study on taste sensitivity to PTC is taken as an example. In their study, they recruited 232 males and 220 females and found that the frequency of tasters and non-tasters among males was 154 tasters and 78 non-tasters, and females 165 tasters and 55 non-tasters. Allele frequencies of taster is depicted with a capital T and a non-taster with a small t, lower caps, and can be calculated using Hardy-Weinberg law, that is p square plus 2p square plus q square. The p is the frequency of the dominant allele in the population, that's the capital T here, and then the q is the frequency of a recessive allele, that is small t. The frequency of tasters, the dominant ones, would be capital T together, that's 154 in males and 165 in females, whereas q square, the frequency of non-taster, recessive TT, would be 78 in males and 55 in females. Therefore, you can see how the non-taster allele frequency is depicted in males and females, and then the taster allele frequency can be calculated as q minus 1. For males, it was found to be 0.426 and for females it was 0.50. Homozygotes and heterozygotes together, that's capital T together and capital T small t and then TT, can be calculated in the following manner for males and then for females. The significant differences between frequencies can be compared and they can be tested for significance through chi-square test as well. Thank you.